So welcome everybody to the April version of IIDR rounds. I'm Lori Burrows. I'm the interim director of the IIDR. It's my great pleasure today to welcome our speakers, Sarah Khan, Andrew McArthur, Kara Tsang, and Ahmad. So I'd like to introduce our speakers. Sarah Khan is an assistant professor and a pediatric infectious disease specialist at McMaster Children's Hospital. She serves as the associate medical director for infection prevention and control and the pediatric stewardship lead at Hamilton Health Sciences. Today, she'll be speaking about SARS-CoV-2 outbreaks in the clinical setting. Andrew McArthur is an associate professor of biochemistry and biomedical sciences. His lab's research program is rooted in bioinformatics, functional genomics, and computational biology, spanning complex informatics approaches to functional genomics of microbial drug resistance, the development of biological databases, and much more. Today, he'll be speaking about the evolving challenges of SARS-CoV-2 in our community. And as I mentioned, we're also joined today by two of our Braley Fellows. So the Braley Fellows program is a program that we have at the IIDR that introduces basic science PhDs into the clinical world of clinical microbiology. So as part of their fellowship program, they are working on this CoV-2 project. So Kara graduated from the Biomedical Discovery Commercialization Program at McMaster in 2016. She's currently a fifth-year PhD student in the McArthur lab. And outside of her PhD work, she helps Dr. Sarah Khan manage clinical research projects. Sherry graduated from the Biomedical Discovery and Commercialization Program in 2017, and he is currently a fourth-year PhD student in the Whitney lab. Outside of his work with the Braley Fellows program, his PhD work has focused on characterizing various mechanisms of interbacterial competition. And today they will be discussing the genomic analysis of the ongoing outbreak of SARS-CoV-2 at the Juravinski. So thank you everyone for speaking today, and I'll turn the floor over to Sarah.
Great, thanks Lori. So welcome everyone. As Lori mentioned, I'll speak mostly in the beginning of this talk about our experience with outbreaks at the JH and some of the lessons that we've been able to learn through our sequencing partners. And then I'll hand over to my colleagues to speak more in detail on that. So there we go. Briefly, we'll start off with just some epidemiology reminders, which I think most people are familiar with at this point, a year into this, as well as our current state. And then I'll shift gears, speak briefly again about COVID pathophysiology and transmission, so that then we can move into a discussion around outbreaks and what we understand around mechanisms or modes of transmission, the concerns around variants of concern, and then shift gears and speak about the Wave 2 JH outbreak experience through the month of December. So just a reminder, this is sort of taken from PHAC, but essentially this is our COVID count list for sort of a 7-day period last week when I took this slide. But of course, this past weekend, we have surpassed 1 million COVID cases in Canada. On the right there, you'll see our Hamilton curves, and you'll see our Wave 1, followed by our Wave 2, and our rise up into Wave 3 now. Reminder again about what the focus and the importance of this disease has resulted in terms of our acute care facilities. So patients hospitalized is one of the big metrics that we all are following, but currently our vulnerabilities is around our ICU bed capacity and those that require mechanical ventilation. And you can see sort of Wave 2 and the rise of Wave 3 just starting here. Again, this data is pulled last week, and so even now it's risen since then. Importantly, it's important to note, who does COVID infect? Well, it really can infect anyone, but the severity of disease is where we're most concerned. And so this graph is very obvious to show that those that require admission to ICU are those that are above the age of 60, and those that have unfortunately passed from COVID have been at the highest risk above 70 and significantly high risk above 80 years of age. And this is really where the focus of the conversation has to be around the highest risk populations. This is also just, I think, comical because it's last week to this week and what sort of changed in Ontario. So this was sort of the framework that existed last week, where there was a more regional approach to our considerations of how we're trying to control this with prevention versus all the way into lockdown and shutdown. And then over the on Friday, this was the change to the graph, became the full lockdown with a white graph across the province. And then today, as we all know, 2 o'clock, we're getting more specific instructions from the Premier. So I won't spend much more time on this talk about the challenge of lockdowns and the considerations and whether that is the appropriate approach, but that's a very controversial topic, and I know there's lots of interesting opinions to read right now. So that was sort of the brief epidemiology primer. Now shifting gears into our pathophysiology and infection side of things. So what's fascinating about SARS-CoV-2 is this viral S protein, which is what allows it to bind to our host ACE2 receptor for cell entry. And we know that the viral load typically is highest in the upper respiratory tract in early disease and replication, and how we typically make the diagnosis using a nasopharyngeal swab. As disease progresses, it can, descend into the airways and get into the lower respiratory tract, where, sputum detection is more possible. And of course, we know susceptibility to infection seems to increase with age, and that may be related to our ACE2 expression being lower in children less than 10 years of age, but this again is still sort of a hypothesis not yet proven. Transmissibility from children also remains sort of this lack of clarity, evolving data suggesting that perhaps it is the younger age children that are more transmissible, whether that's a true pathophysiologic phenomenon or just an epidemiologic reality, in that children need much more close degrees of care and are less in control of their respiratory droplets and secretions. But again, evolving data, as a PEDS ID person, this is sort of more of an interest to me. And the other sort of interesting point that we'll move into as we talk about variants of concern is how RNA viruses are rapid in their ability to mutate, and thereafter leading to this concern of variants of concern. Again, there was lots of early debate around and remaining debate around transmission mechanisms of SARS-CoV-2. And I'm briefly going to summarize sort of the debate and concern, because it still comes up when we're dealing with outbreaks in certain facilities that may be older or may not have the same degree of air exchanges per hour that we typically have in our acute care facilities. So again, the early days, we really dichotomized respiratory viruses into droplet contact modes of transmission versus aerosol routes of transmission. And, there's variables that we all look at that help us understand, which side of the fence anything sits on. I think SARS-CoV-2 has really challenged that dichotomy and made us truly recognize that it is more of a spectrum. And it depends on the conditions one is concerned about around transmission. Some of the sort of more basic arguments one could make around this is that the low R naught that we've seen with SARS-CoV-2 as compared to truly airborne pathogens such as measles is sort of one hallmark that one can clearly recognize and see in the degrees of transmission we see with things like measles. The other reality being that the majority of SARS-CoV transmission has been around short distances rather than the long distances one would see with airborne transmission. Though, of course, droplets and aerosols could be occurring at a short distance as well. Perhaps the inoculum is not achieved at the farther distances that it would be required to actually result in infection. The other sort of argument that's made in acute care facilities is, for the most part across Canada, we've been managing COVID with droplet contact precautions aside from aerosol generating medical procedures. And for the most part, this has been effective in preventing transmission throughout our acute care facilities. Again, there's interesting RCTs in progress. Dr. Mark Loeb from here at McMaster is the PI of this clinical trial that's underway now. But again, that will be sort of real world data to help us truly understand what is the most appropriate form of PPE we should be using. In terms of air sampling studies, there's lots of in vitro data that have demonstrated viral RNA that can be aerosolizable. This virus can definitely be aerosolizable under the right laboratory conditions. And that has been demonstrated much through lab conditions or even air sampling studies where viral RNA is present, whether that's culturable and viable virus that could actually infect another. That data is still somewhat less clear. And then the other sort of case reports around super spreader events or potentially super spreader people themselves that may have shed at a higher viral load, some of this relating to a dispersion or a K factor. Much of this is probably, again, dependent on the environment one is in, and the activities one is engaging in. So there was a highly published prior practice where there clearly was transmission beyond 2 meters, and that's been cited as sort of data or proof that that is an aerosolizing, an airborne transmission mode. There's been conflicting reports of other settings, again, community settings. We're not talking about acute care facilities with the air exchanges per hour and the differences that we have in hospital type setting. But for example, in buses, this is another sort of case report that was published around in red, there's the index patient and then the degree of spread that occurred in a bus. Now, again, the ventilation in a bus being very different than what we would see in an acute care facility, but there's conflicting data around flights, which similarly wouldn't have the same degree of air exchanges per hour, where the degree of transmission was not in keeping with what one would see with airborne, but more in keeping with droplet contact. So all that to say, lots of evolving evidence, lots of different folks from different sides of the coin here, aerosol scientists, clinicians that have debated this much in the literature and ongoing continuing discussions happening. The other point around any droplet contact pathogen is the possibility for respiratory droplets to be on the local environment within sort of 2 meters and the risk associated with that. That is why we use droplet contact precautions in hospital settings. True fomite transmission truly, in early days was this concern, but hasn't really played out in terms of real modes of transmission. So I think for the most part, people aren't wiping down the groceries the way they previously were. I won't get into discussions around zoonotic transmission, the rare case reports of vertical transmission, again, in typically immune compromised women with severe disease at the time of delivery. And then again, there's theorized discussions of sexual, fecal, oral, and bloodborne transmission, but truly not yet documented. So when we talk about the transmission period of SARS-CoV-2, which is really important in order to understand how we manage outbreaks, again, the bulk of transmission occurs at that prolonged close contact. So households being where you see high secondary attack rates compared to more community settings, schools, and even in hospitals. Limited transmission to those that have transient exposure or again, contact at distance. And we've talked a little bit about the opportunistic aerosol transmission in crowded or poorly ventilated indoor settings, not really what we would see in acute care facilities. Of course, the other key aspect of managing an outbreak is understanding the incubation period of the pathogen at play. So there's lots of good data now confirming the median incubation period around 5 days, but symptoms being able to onset anywhere from 1 to 14 days post the last exposure. And then the other important part of understanding transmission is the infectivity period. So we know that one can be infectious to others 2 days prior to symptom onset. So there is sort of that pre-symptomatic infectious period, followed by typically around 10 days post-symptom onset in most mild to moderate disease. There is the possibility of residual transmission that could occur even longer after, even beyond day 10, but that's typically only seen in those with severe illness or those who are truly immune compromised that may be shedding for longer and do not have the immune response that the majority of the population would. So now shifting gears into outbreak management, Ontario now has sort of more clear-cut definitions around what we define as an acute care facility outbreak. So that does require 2 or more lab-confirmed COVID-19 cases. Those could be within patients or staff within a specified area, such as a ward or a unit or a floor, within a 14-day period. Typically, you have to sort of have some degree of reasonable rationale to say that the cases were acquired in the hospital and didn't come in with it and were incubating upon admission. The other factors you would consider are how many confirmed patient and staff cases are currently on the unit, how many are symptomatic or just asymptomatic, what was their duration of admission before they onsetted their symptoms or turned to have a positive test, because again, that might help you understand maybe they got admitted with it, were incubating, and it was truly a community acquisition and not an in-hospital acquisition. If there's any known prior test results, again, because if someone had COVID months ago or weeks ago, they may still be testing positive and you don't need to be concerned about that positive test at this time. It's residual, likely dead RNA. And any known exposures in the community or in the healthcare setting that can truly help you create an epi-link and understand the mode of acquisition or how that transmission occurred. Now, outbreaks, as I've mentioned, can be patient-to-patient transmission, and that's how it can perpetuate. Sometimes the index case is known, and oftentimes it remains unknown. Sometimes there's a clear pre-symptomatic visitor that the next day you find out, oh yes, the father was visiting, and the next day they're now symptomatic and testing positive, and then you have a clear epi-link as to how the exposure occurred while in hospital. We also have had challenges with staff-to-staff transmission. Some of that occurring, in break rooms on a ward where people have to unmask to eat and drink. Otherwise, facility-related outbreaks can occur just in shared office spaces, again, where people may not be as cognizant of their PPE practices, or even outside the facility where, co-workers are carpooling together, or they live together, or just socializing outside of school, and then the two cases end up getting linked to a given hospital ward. And then there's always the possibility of just coincidental with rising community epidemiology to community cases that are, picked up in the community, but end up working in a single area. And then you're sort of stuck without a clear community epi link that, oh shoot, is that a transmission that occurred on our ward? So that's where staff-to-staff outbreaks can be a bit more challenging. So why is SARS-CoV-2 so much more complex in terms of a management of acute care facility outbreaks compared to, what we used to deal with, rhinoenterovirus or influenza? Well, because of that longer median and maximum incubation period than most of our respiratory viruses, it really can delay our recognition and then our ability to control an outbreak. And as I mentioned, that pre-symptomatic phase of transmission really gets you behind the 8 ball in terms of trying to get things under control, because there's already transmission occurring before you even know a case is a case. And then the other challenge with asymptomatic transmission being possible. And that really is likely less of a risk of transmission compared to a pre or a symptomatic person, but it's still possible. And with vaccine, we may actually see more of that possibility occur. And then the lack of population immunity, again, the degree of sort of disease that could spread rapidly in a completely non-immune population makes it a higher risk for you to actually end up with outbreaks. And then on top of that, as we've had more and more cases and experience with it, the challenges of finding remote infections and having positive tests that actually aren't reflective of active disease can also throw you a little bit of a curveball, because you have a positive test and you may not have a history, but it may be an old burnt out infection at this point in time. So what do we typically do to try and prevent? Well, the goal would be to have surveillance, active surveillance, every single day. Every patient and staff is assessed to see if they have any symptoms, isolated should that occur, and tested as quickly as possible to know if you have a case. Once a case is confirmed based on testing, then you have to pursue the contact tracing, typically for the 48 hours prior to their symptom onset, as we mentioned, that pre-symptomatic period. But if they are actually asymptomatic and testing positive, we typically go 7 days prior to that positive test status, again, to sort of see possibly where transmission might have occurred in that interval period. And then the other challenge is trying to understand, was this acquired in hospital, or is there another epi-link that can confirm where this case came from? So what we do when an outbreak has sort of thought to have been occurred or have occurred, all symptomatic or known positive patients are put in isolation. So ideally in single rooms, but in some of our older hospitals, that's not possible. We have 2 and 4 bedrooms from time to time. Or you can alternatively cohort them with other confirmed positives, because then you're not putting more people at risk. Any positive staff, of course, are sent home in self-isolation. And we do active surveillance for any symptomatic staff or patients that may be coming up. And we try to cohort the outbreak exposed staff away from wards that have not had outbreak exposed staff. But even that's a challenge, because there are some clinicians or healthcare providers that do need to go throughout the hospital just based on the service they provide. Exposed patients are also put in isolation because they, again, could turn positive after their degree of exposure to a known case. And then high-risk exposed staff are typically also sent home, again, in self-isolation because of their risk of converting to a case. We try to minimize any group activities, so group meals or exercise or other things that are part of hospital care, we often have to sort of shut down, again, to reduce the risk of transmission. We typically close the wards to admissions or transfers into the unit, which creates huge bed capacity challenges. And that's really what was one of the big challenges at the JH in December. Anyone who was transferred off of that unit in the last sort of 48 hours prior to the outbreak being declared are typically sort of put in isolation wherever they've landed, if it's at another facility or in another ward, and we often test them as well as they're part of that outbreak cohort in the end. And then we typically discuss the need to restrict visitation, recognizing the importance that visitors play to our hospitalized patients, with the goal to really restrict traffic to the unit, again, to mitigate risks of transmission. We have lots of meetings as the outbreak management team, and those include multiple stakeholders, including leadership, public health, infection control, occupational health, and of course communications both internally and externally. We review the data that we have to date, the case and contact tracing information, as we're implementing our control measures, trying to understand a root cause, and making sure our PPE are cleaning, and we have good communication with our stakeholders inside and outside of the hospital. And when can we declare it over? Once we've interrupted transmission, which is typically defined as no further staff or patient cases occurring for 14 days since the last onset of symptoms of the last positive case. Or if it's a healthcare worker, the last shift that they worked when they were potentially infectious. So what is going to be the challenge with the VOCs? Well, the concern is that as these RNA viruses are mutating and adapting to us as a human host, they are picking up these mutations, which make them more able to bind to our receptors, reducing our ability to develop antibodies to sensitize or basically evading our immune system, and also the potential to sort of replicate with higher viral loads in the upper respiratory tract, therefore making us more infectious as hosts. And so there's sort of the three main variants we'll talk about with their shared mutations. I won't go into the specifics there. So just to speak to how this challenges outbreak management. So we know that the UK variant B117 is at least 50% more transmissible. This is also likely to be seen with the Brazilian variant, less clear right now with the South African variant. In terms of severity of disease, emerging data now suggesting that the UK, likely the Brazilian variant, are going to cause more severe disease. And for the most part, it does seem that our PCR tests with multiple targets should still be able to detect these. And so we likely will be okay in terms of diagnostics. Around our ability or our immune system's ability to sort of still retain protection against a potential new variant that may arise. The concern being that the Brazilian variant did emerge despite a 75% seroprevalent population. So that's going to be a real concern whether, those with past infection will still remain immune to potential new variants. So this is going to be a significant challenge, and even further concerns around whether they will have immune escape from our vaccines. And again, early days in vitro data showing that the mRNA vaccines will likely be able to protect us from the B117. Still early days to say around South African and the Brazilian variant at this point in time. In terms of what we can do in hospitals, we can cohort B117 with our local strains, and that's been really helpful, and that thankfully has been our main strain or variant of concern at this time. Though right now we are not able to cohort our Brazilian or our South African variants of concern with any local strains, and that is going to put a significant strain, again, with the risk that we likely won't have we will have risk of reinfection that might occur with these variants, even if someone has had past history of SARS-CoV-2 infection. So just to speak briefly to VOCs, BC and Alberta is seeing B117 in significant numbers. In Ontario, we're starting to see the rise of all three, and Quebec, mainly just the two there. So now, in the remaining time, we'll talk more about our Wave 2 experience at the JH and the outbreak that we saw. So just to summarize the COVID cases we've had admitted to hospital, we had about 1,000 over the past year. 6% have passed. The mean age was 63 years, and a very small minority were pediatric patients. This is sort of the wave data that correlates with our community prevalence, and this is sort of how we're defining our three waves. And in terms of the waves and the bulk of them being community acquisition versus nosocomial, wave 2 was again majority community acquisition, still seeing long-term care outbreaks. As we're entering into wave 3 at this point in time, we're actually seeing more nosocomial transmissions occurring within hospital as opposed to community acquisition. So again, the outbreak I'll be focusing on is our December 2020 experience, which I have more data on the patient side of things, so 33 patients, but we also had 48 staff cases as part of that large outbreak that was across our JH hospital. So in terms of that sort of December period, the 33 patients ranged from 48 to 94 years of age with a mean age of 80. The outbreak sort of lasted December 3rd to 22nd, and the bulk of it being on our E and F wings of our hospital, 80% of cases occurring there. We had a couple of cases on B3 and M2. This is sort of the actual epi curve and the onset of dates, and you can see it was clearly a propagated outbreak. It didn't sort of have a peak and a fall. It really continued to propagate. To remind you a little bit around our landscape here at the JH, the E and the F wards, again, were our most troublesome wards. They're quite old and they were renovated, but part of the challenge being these are multi-bed rooms, which also perpetuates the risk for easy transmission to occur. And then our B3 and our M2 wards were actually less affected, so I won't speak to that in much more detail. This is interesting, I think, because it shows you are in green where our B3 sort of cases occurred and went up and came down, really just in blue there. Our E3 outbreak was really quite prolonged. It lasted us most of the month, and that's in green. Our F5 outbreak also sort of was these sort of perpetuating cases that continued really throughout the month. E4 was sort of a brief up and down curve in beige there, and then M2 was just a couple of cases towards the end of the month. So what was interesting that we were able to get back from our sequencing partners was to understand a little bit more about what strains we were seeing. And so it really was this purple wave that crossed over JH. And this was the B1369 sequence or strain that seemed to really be the bulk of our cases, with the occasional community case, another importation seen there, a couple of other strains. Now, what I think is fascinating about this is that this strain arose in China a year ago in March 2019, March 2020. But what's fascinating is when it was peaking in December or January, that was the Canadian peak, and then it moved to the US and has continued to cause disease there. But it sort of timed exactly with when we were seeing our challenges at the JH. So this purple wave that went through the JH, again, just to see the breakdown of where that occurred, it really was throughout all of our wards. And again, with the focus being on the E and F wards mainly. And again, as I said before, there was a couple of other strains that were found. Those were likely just community importations and not really the big culprits that we saw. This one, the blue strain in here, B1, was likely a community import, but clearly did have transmission occur within ward to ward. But what I'll focus on is the B1369 and sort of show you how it went through and trekked through our wards. So again, this is the cases broken down with the patient cases at the top there versus the staff cases. And so there were sort of staff cases grumbling along. They peaked and then settled, but the patient cases just continued. And that likely relates to sort of the potential that staff contributed to potentially to some of our patient cases as well. I don't have a lot of the ward attribution, like which staff came from which ward, but of the 14 cases, again, it was mostly the E and F wards that were seeing clear staff attributions. So just to follow Ward B3, this was our short 2-day sort of outbreak in terms of the numbers of cases, though obviously it lasts longer before you can declare it over. Again, it was B1369, 3 of the 3 cases, random cases across the unit really, and then a neighboring next door room also got infected the following day. And that was the extent of the B3 outbreak. Our next ward here, E3, was, as I said, kind of went through the whole month essentially, and 9 of the 9 cases were B1369. And so it's interesting to watch it go through our unit here. So sort of sporadic cases occur across and diagonal from the unit, and then you have it just moving from these room to room. It's not sort of following a line or transmitting sort of specifically room to room. It's random sporadic cases as it goes through our facilities. And then this was also interesting to see that the patient cases were sort of bridging around the staff cases. And so likely this sort of started on the patient side, went to staff, and then settled. Our E4 ward also sort of was a busy middle of the month week where we had 7 cases, 6 of them being the B1369. But what's interesting here is the first two cases on December 10th, one was a community import clearly, and the other was our B1369 that got transmitted to this ward. But because we found 2 cases and we don't have sequencing data up front, we declare an outbreak. And that allows us likely to catch the subsequent cases faster because of the degree of active surveillance and control measures. And that may be why we were just sort of able to abort this within a week's time. And again, the staff cases mirror the patient cases here as well. The next ward, F5, December 4th to 20th, 9 of the 11 cases were B1369. And again, we have 2 community imports in the early days, but then B1369 spreads pretty quickly. And again, it's random, it's across the wards. It's not sort of following a sequence room to room the way an airborne pathogen typically spreads. And then we sort of, eventually it burns out by December 20th, and the staff cases are sort of closely aligned with the tail end of the patient cases. And our last ward is M2, where we had essentially sort of the single case that was a community import, but likely allowed us to detect these other cases relatively quickly. And it was sort of 2 of 3 cases that were our B1369. So take home points, what did sequencing help us understand and sort of address? And what hypotheses did we confirm or deny? These were not just different multiple importations, which was a question. Are we just seeing a rise in community prevalence and then we're just seeing coincidentally cases? No, there was clearly a purple wave that went through. We were able to confirm it wasn't a VOC. That was the concern given the degree of spread that we were seeing. It was a shared strain, but it wasn't one that's known to be more infective. Overall, I would say that this is sort of reassuring that we weren't seeing airborne spread. It was random cases on our spot map and likely sort of associated with roommates or clusters of cases around an area. We never were able to sort of find a typhoid Mary, a single link that attached all of these cases on any of the specific units. We couldn't identify a piece of shared equipment that was the specific concern. And for the most part, one could argue this is not a failure of PPE. There was no evidence that we needed airborne PPE. Droplet contact PPE continued throughout, and eventually the issues did settle. We didn't have to change practice specifically. And staff were donning eye protection for outbreaks, but it didn't seem to prevent transmission on units to staff. And it sort of brings us to the conclusions that there's likely some degree of micro breaches that are occurring, and it allows for patients to staff transmission and vice versa. So that's where I'll stop and hand over to my colleagues.
Hi, everyone. So what Cara and I are Braley Fellows, part of the IIDR, and what we'll be talking about for the second half of this talk, and then Andrew will follow, is about phylogenetic analysis of the Juravinski outbreak samples. So I think everyone here is probably very familiar with the importance of genomic sequencing. Sequencing, I think now more than ever, has become much cheaper and more efficient. It's no longer, I guess, the end of a project. You know, it's not the final goal. It's actually something that is done routinely. And so when it comes to something like COVID, it's, you know, as simple as just taking a small sample and sending it to a sequencing facility to determine its sequence. And this has been instrumental for a number of important healthcare advancements. So when it comes to the SARS-CoV-2 genome, just a little bit of molecular biology, I guess. The genome is about 30,000 base pairs. It's an RNA virus, so this is an RNA genome. And the complete transcriptome was elucidated last year. There are 10 main genes that encode both non-structural proteins and structural proteins. So ORF, just as an example, ORF1A, constitute a majority of the SARS-CoV-2 genome and are encoded for important non-structural proteins like the RNA polymerase and viral proteases. And then you have the spike protein, as well as the nucleocapsid protein and envelope protein, as Dr. Khan mentioned, that are important for virulence. And this is just the virion structure here that resembles classic coronavirus structure. And so overall, the sequence was incredibly important for a number of downstream applications. So that includes diagnostics development, vaccine production, as we saw especially with these mRNA vaccines that have been recently developed, and currently, I guess, the biggest thing, and also what we're interested in, is detection of new variants. So when talking about variants, there's a lot of different, I guess, terminology used. We talk about them, I guess, as lineages. So essentially, a lineage is, of course, just something that arises from an ancestor. But to define a lineage, there needs to be a cluster of sequences that appear novel and coincide with an epidemiological event. And then these can be analyzed using phylogenetic analysis and compared to the ancestral sequences to determine evolutionary relationships. So when defining a lineage, it again has there are a few key requirements. First is to form a clade, or just a group that differentiates it from the ancestor sequence, and then epidemiological evidence. This is important, obviously, because you need to have some evidence that this is causing infection and circulating through a novel geographic region. And lastly, there needs to be defining mutation or mutations that make this lineage unique. And so when we were given the Juravinski Hospital outbreak samples, it was for the B1369 lineage, as Dr. Khan mentioned. So there were a total of 76 positive samples that were received from the hospital, 55 of which were of this B1369 lineage. And the rest on this table here, the B1 all the way down, these are likely community acquired and not part of the outbreak. And so when we were interested in doing this analysis, we wanted to compare these sequences to known sequences in the GSAID, or GIS-AID database, which is a repository of COVID-19 sequences that has accumulated, I think, up to 1 million sequences over the past year. And it's an open source database for individuals who are interested in comparing sequence data for COVID-19. And so we wanted to compare to that. And we also wanted to add in or incorporate metadata about the patients. And so there were a total of 36 patient samples, or 36 patients that we had metadata for, sorry, there was a total of 36 total patient cases. The rest were staff, and then we had metadata for 92%. And so the average age was 81 years. And then there was about an even split between male and female cases. And then we also knew the time at which the patients tested positive. So when we were, our first goal was to construct a phylogenetic tree that compared the Juravinski outbreak samples to the ones in the GIS-AID to see if there were any major differences. We weren't particularly interested in finding specific differences. This, I will go into later, is a little bit difficult with the current sequencing data, but we were interested to see major trends. So we used a maximum likelihood tree. And this is just a computationally intensive method of generating a phylogenetic tree that is based on probability, and it essentially finds the most suitable tree based on the likelihood that all the data fits, or all the sequences fit the tree construction during its assembly, and it's the gold standard for phylogenetic analysis. So when we generated this phylogenetic tree, again, we used the GIS-AID sequences for the B1369 variant. And as you can see in red, they all cluster together very nicely. And to our surprise, the Juravinski outbreak samples actually form their own cluster at the end here in blue. And we wanted to focus more specifically on these strains and incorporate metadata. So this is just a zoom in of the of that region of the tree. So first, what we did was we incorporated staff, or we wanted to differentiate staff versus patient cases. So in this modified version, the darker circles represent staff cases, whereas the moderate blue is our patient cases, and then the very light blue are unknown. So this is just an example of the metadata that we incorporated. So we wanted to incorporate date. So this is just a, I guess, dummy node and spot here that, just as an example. And so we wanted to incorporate date, the unit and room number, as well as genotype. So for date, we used this gradient, which yellow defines the earliest time points, blue defines later time points. And what you can probably appreciate is that there's no real trend, no easy to identify trend that occurs in this data set. There's a mix of yellows and blues across the board, which was a little unfortunate, but I think as I will go on to say later, it's a little bit difficult with our current analysis to really pin this down. But we also included unit number and room number. And so, so the colors here denote which unit the infection or the sample was retrieved from. And E3, E4, and F5 all appear very close to one another and in the hospital, and do constitute the majority of cases, as Dr. Khan also alluded to. And so it is, I guess, important to, or it's good that we're seeing that trend. And then we also included genotype. There are 5 unique genotypes within this, I guess, variant. And it was mostly, most of the samples were consisting of either genotype 2 or 3, which were very closely related. And that seemed to have been present in most of the patient samples. So there are, while the metadata does provide us with kind of an overall trend that, yeah, E3, E4, F5 are the major, I guess, areas where infections were occurring, as well as the lineages that arose from the ancestral B1369 variant do seem to also come from those wards. So there's, I guess, some general trend that we can find. It's still difficult to really pin this down because the data is, one, we don't have metadata for staff. So if we had that, that could also maybe help us identify more important trends that maybe we missed with our current metadata sample. Of course, if we try and do this analysis on more patients, maybe more trends will start to come up. And then just a small thing about the clustering here. I mentioned at the start that the order in which the sequences appear isn't necessarily indicative of their evolutionary, I guess, trajectory. And so some of the sequences that appear earlier may not actually be there. These are very closely related isolates. And so the order and placement of these different sequences in this tree is highly dependent on the sequencing resolution. And so if the sequencing data is a little, or isn't, if the depth isn't high enough, then we're going to miss some more data. And we may not be able to find those exact differences in the order of sequences in this tree. Okay, so Cara will take over now and talk a little bit more about our downstream analysis that we did.
Great, thanks, Sherry.
Yeah.
Okay, so next we wanted to see if there was another method of building a tree that would provide perhaps a different interpretation of our data. So instead of using maximum likelihood to build the tree, we are now using an algorithm called minimum spanning. So this method is a set of connections that link together samples by minimizing the distance, and it is less computationally intensive compared to our previous algorithm, and it is typically used for clustering similar things, and in this case, it's sequences. However, there is a limitation in that this algorithm builds one path and one path only, and so building this unique path makes it blocks different alternatives that could potentially be more direct and alternative to what we see here. So in this tree, we have our Juravinski samples as well as our GSED B1369 samples. And so in the circles represent the proportion of samples that are proportional to the number of samples. And so the bigger the circle, the more samples that are represented. And so as an example, in red, we see this one GSED bubble, and that signifies that there are many samples that have similar or the same sequences. And so what we can, and so the distance between each of the samples is the genetic difference between each sample. And so, for example, if a connection was the length of the bar at the bottom, that would indicate there is approximately 5% of genetic difference between one sample and another. And so the takeaway from this tree is that Juravinski samples are very similar to one another, which is why they cluster together. And there are also a few GSED samples that are also clustering either within or just right beside this Juravinski cluster. And so this suggests that samples that we have seen at the Juravinski outbreak have also been seen elsewhere in Canada as well. Next slide, please. So up until this point, we did all of our analysis in February 2021. And so over the weekend, we wanted to refresh ourselves and learn more about this B1369 lineage. So we re-uploaded our samples into Pangolin, which assigns lineages. And we found, to our surprise, that the B1369 lineage samples were now reassigned to a new lineage called B15641. And this lineage is a Canadian lineage, and the parent lineage is from the US. It's B1564. And this lineage was split from the previous lineage assignment called B1369. And so here we have the geographical distribution of the sequences that have been seen around the world so far. So most of them have predominantly been seen in Canada, which makes sense as it is a Canadian lineage. And one sequence was found in Australia. And then in the next graph, we see the number of sequences on the y-axis, as well as the timeline at which these sequences were found. And so the earliest date of this B15641 lineage sample was found in August 23rd, 2020. Next slide, please. So what we wanted to do was recreate the trees that we previously created with this new information. And so with this maximum likelihood tree, the previous one, as we can see, the Juravinski clade groups to the edge of the tree. And so at that point, we thought maybe it could be a new lineage because it doesn't really group within this B1369 lineage. And so we recreated this tree with the GSED B15641 lineage. And now we can see that the Juravinski samples actually cluster really nicely within this lineage. And so this suggests that the Juravinski outbreak samples does belong in this lineage. And of course, with more information and potentially more Juravinski outbreak samples, they could potentially form their own lineage, but we would have to have more information and more sequencing data. And then next, we decided to recreate our minimum spanning tree. And so again, here we see that the Juravinski cluster groups really well together. And again, we see a few GSED samples that sort of linked together with that cluster, suggesting that these samples have been seen before in Canada as well. Next slide, please. And so looking at that from a national level, we also wanted to see where our samples were in the global context of the pandemic. And so we've talked a lot about lineages, but there is also clade nomenclature. So a clade is essentially a higher order or level of assignment, and it is a group of lineages essentially. And so currently around the world, there are 11 different lineages, 11 different clades, and each clade is defined as defined by a signature mutation and are named once the clade reaches greater than 20% global frequency for 2 or more months. And Sherry, next slide, please. So each of these colors represent one of the 11 clades that are found around the world. And in the middle, we see our Juravinski samples grouped within 20C. And next slide. And so here we can see that it's a zoomed in version of the tree, and our samples are clustering very well within this 20C clade. Next slide, please. And then this is a global map distribution of the 20C clade around the world. The bigger the green bubble, the more sequences there are in that particular country. So you can see it's very widespread. And so Sherry and I would like to summarize what we've learned as Braley Fellows. With the help of our clinical colleagues and our bioinformatics expertise, we were able to further interpret the Juravinski outbreak using phylogenetics. We realized that our results are limited to the sequence quality and depth, and so perhaps using deeper sequencing would increase our tree's resolution. We learned how dynamic and crazy the pandemic is, where new data can change the narrative around a cluster of sequences, as we saw that our samples were reassigned to B15641. And we also learned the importance of open access sequence databases as well as open source tools, as we would not have been able to do any of this work without the tools that were easily used and very informative online. And lastly, we would just like to thank our fearless leader of the Braley Fellows program, Dr. Dawn Bowdish, for giving us this opportunity to take on this project and really learn so much throughout our time as fellows. Lastly, thanking Dr. Sarah Khan, Dr. Al-Jarbu, and Dr. Al-Salem for their clinical expertise, and Dr. Andrew McArthur, as well as Jalise, for their bioinformatics knowledge. Thanks so much, and I'll pass it off to Andrew.
Okay, in 2 minutes. So what's going on in our community? So we've heard about one outbreak, but what is the data in our community? So quick review. The first batch of data we had at the genomic level was in June 2020, so right after the sort of the first wave. This was an LTC and hospital outbreak with movement of patients between. And this outbreak was what was called the B11 lineage, which was one of the early lineages that spread around the globe. And so we knew that was in Hamilton, but we didn't get any community data. So for all we knew at that stage, that everyone in Hamilton that was positive could have been B11. Diversity was very low at that early stage of the pandemic. The next glimpse of data we got was in November 2020. With Public Health Ontario, we did a point prevalence study. So we tried to sequence everyone that tested positive in a 24-hour window on November 24th. 80 of them sequenced. What we found is at that time, there was a lot of diversity in Hamilton, 19 different variants, 5 of them predominant. So we knew that there was a lot of that. Hamilton really didn't reflect a one importation event, that it was quite diverse in our community. Next set of data that became available was what you just heard about, the Juravinski outbreak, which was the B1564.1. But what was noticeable, we also saw 7 other variants, so evidence of community transmission. So this is outbreak data, but those 7 tell us that there's still lots in the community when it comes to genomic diversity. Now, this is about the time that the variants of concern started to come out. So the prediction is that over time, things will become quite homogeneous, as it'll be the 117 or the 351 that take over. So Public Health Ontario did a point prevalence study on January 21. The data actually, it is public sequence-wise, but they haven't geolocated it down to city, only province. So I can't tell you what lineages were in Hamilton at that time, but I can tell you that we did find B117s in our community. And then in February, we had an outbreak in a vulnerable community that I can't really give you a lot of details about, It was a rapidly escalating outbreak, so we really expected a variant of concern. And what came out was this variant called R1, plus 3 other variants. So there was a little evidence of community diversity, but this R1 variant literally had only been named 10 days before. And what was striking outside the concern for these members of our community, that Public Health Ontario designed a quantitative PCR test for the 501 mutation, and it was negative for that. So we knew it wasn't a VOC. And it would have just flown under the radar at this point, except for Merrick and his crew at St. Joe's created an quantitative PCR assay for the 44K mutation, which is known in 2 out of the 3 VOCs, and it came back positive. So we knew it wasn't a VOC, but it had this one mutation associated with increased transmission. And so we rapidly worked with them to sequence, and what we found is this R1 lineage, and it had this additional mutation at the furin cleavage site. So there are now increasing number of lineages around the globe that have the 44K and some sort of mutation near the furin cleavage site, all associated with rapidly escalating outbreaks, but no good biology behind it. So they are considered variants of interest. But what was really great, we got the sequencing result over the weekend. We contacted David at St. Joe's. They immediately went back, did fresh swabs, and I can confirm that the Mossman and Mubareka labs now have live virus of R1 in the BSL-3 facility. So with luck, we might be one of the first institutions that actually test these new, increasingly frequent variants of interest that have these furin cleavage site mutations. So what about now? The key thing is that we have these 2 quantitative PCR screens. So the data you see provincially and locally really comes from tracking individual mutations. 501Y is 100% correlated with being a VOC. But also St. Joe's is now a spoke in the Public Health Ontario network, setting up their own sequencing and surveillance facility that we'll be partnered with. And the goal is very soon to sequence 250 variants of concern per week based on RT-PCR results, as well as 250 samples from the community that are non-VOCs. And so this engine, once it's up full, will be Hamilton and Hamilton surrounding areas and other communities. This will really give us a great sense of what's happening in our community and whether diversity is indeed decreasing and VOCs are taking over. And I guess that last bit, why this is really important, you need to really think broad about who our community is. Essential workers, people in shelter communities, migrant workers. It's April now, so this is going to be a concern for Niagara Hamilton area as people come in, say, from the Caribbean, and as well as our neighbors in 6 nations. So we really need this surveillance systems to go up, because it's not just the VOCs anymore, but these rapidly appearing VOIs. And as we provide experimental data, I'm sure some of them are going to become VOCs. And to close, just quickly thank everyone involved and all the folks at the bottom that have been doing this sequencing to date. Perfect. Thank you. There you go. Quick, Lori.
Thank you so much to all of you for continuing to work really hard on this evolving, no pun intended, situation. We have time for a few questions, and while people type them in, I'm going to ask a couple myself and take the moderator's privilege. So we keep hearing about how, over time viruses evolve to become less pathogenic, right? Especially coronaviruses, but these seem to be becoming more pathogenic. So can you speak to that?
Dr. Khan, would you like to try that one?
I think the crystal ball is what we're all waiting to see. And I think, natural selection would state the opposite of that hypothesis. I think, things remain to be seen. This pandemic is very different than what we saw with SARS-CoV-1. So I don't want to make any predictions, but I think the fear continues around whether immune escape will continue, and regardless of what we do and what immunity we build or what vaccines come out, we may not truly achieve full control. That's as far as I think I can go.
Yeah, and
I think it's numbers, right? In outbreaks or smaller scale global events, you only have so many lineages in evolution can follow. But now we have so many lineages, so much numbers, that gives so many routes for the virus to mutate in a way we don't like. And that's the B117, for example, has picked up the 44K in a subset of the UK. So now it's got all the mutations that we're concerned about. So that's drive numbers down. That's the only answer. Yeah,
I guess the immune escape thing is the scary part, right? Martha said, so it will become the 5th circulating coronavirus. Are any of you aware of vaccine modifications to account for these new variants that are already underway?
So Moderna has made modified vaccines where the mRNA vaccine expresses the spike protein of each variant of concern. And the early data out for those trials that it's effective. I don't think we're going to deploy them because the data for the existing vaccines on effectiveness against the variants of concern is that we don't have worries yet. They did that in under 3 weeks. So that's one of the advantages of the mRNA vaccines is that retooling capacity.
Go science. I love it. Okay, we're out of time and I don't see any more questions coming in. So I just want to thank all of you again for this fantastic talk. I think it's amazing what we're able to do now with sequencing, that we can keep track of these things almost in real time. I don't know how helpful that data is in actually dealing with it, because I don't think it really changes what you have to do, but it really is important to know if it's changing under our noses. So thank you all, and thanks to our audience for joining us today, and we'll see you next month for May's rounds.